Okay, alright. So, hello, dears. And hello. Okay, so, medyo kumalma na ako. Okay, but I'm still quite, kinukutas. Hinihingal, pero laban lang. But anyway, alright. So, for this second video, no, on our lecture on the preparation of fecal smears, we'll start first again, or we'll go now to the different methods in preparation, okay? And we'll start first with, again, the most popular and the most common and again, one of the methods in the laboratory for parasitology, especially when you work as medtechs na, the most common that you perform good for stool specimens is to create the direct fecal smear. And then we go now to the D'Antoni's method after, and then the Catothic, and the Catocats, okay? All right, so we'll start first with your direct fecal smear. Now, what is its principle? It's a direct wet mount, or other name niya, generally, the direct fecal smear is also known as your direct wet mount, okay? But it's used interchangeably, but your wet mounts, again, can come in different forms, all right? They usually vary in the reagents that they use, all right? So for the first type, again, direct wet mount or direct fecal smear is usually to detect motile protozoan trophozoites, okay? And to determine cellular morphology. So it's to determine or to examine variety of parasites from protozoa to helminths, okay? But generally, it's used so that we can look at motile trophozoites, okay? Because it's under this preparation that we can see them, okay, in the trophozoites that are motile. And aside again from trophozoites, you can also see cysts, you can also see mga helminth eggs, and even larvae of helminths, okay? All right, ayan. Now for the materials, what we need are yan. As you can see, stool sample, of course. What will we examine if we don't have a stool sample? Cover slip, glass slides, applicator stick. An applicator stick is you could use a toothpick, all right? But there are still applicator sticks that are much thicker, no, in terms of the diameter, then it's much longer then. Basta it's much thicker na pagka-stick. All right, okay, that's applicator stick. And our reagent that we use is the NSS, or 0.85% sodium chloride solution. NSS is also known as, again, your normal saline solution. Okay, so sir, normal. So na po yung abnormal, charot. Normal meaning it follows the principle of normality in chemistry. I forgot na the meaning of normality, but yeah, that's the point. Normal saline solution. Parang it talks about the concentration of the saline. Again, 0.85. Some references will say that it's 0.9%. So, gi-round off lang ang 0.85. Alright? So, again, NSS or normal saline solution. How do we prepare that reagent? NaCl powder, 0.85 grams, and your distilled water of 100 ml. So, 0.85 over 100, so that's 0.85%. Okay? Alright. Tama ba? No, Mark. Basta, that's it. Okay. 0.85% of... Mark, yes, atong mga math dyan. Okay, anyway, 0.85% NaCl solution or 0.9 or your NSS, the normal saline solution. Okay, so that's the reagent that we use. All right. But again, as I've mentioned in the previous video, because again of potential errors, no, especially in the transport of the specimen to the laboratory of fresh stool samples, some laboratories, I think in the other countries yata, have forgo, all right, or have eliminated the preparation or the examination of your wet mounts, okay, or your direct fecal smear, because again of possible errors that can be encountered. That is why they now resort, no, or to using preserved stool samples. And once they receive preserved stool samples in the laboratory, they then the samples undergo concentration. So the different methods of concentration techniques, all right? That's our next topic. Okay, all right, ayan. All right, so again, that's the principle and materials. Now, we'll go now to the procedure. Ayan, guys. I hope you appreciate my procedure. As in, ako ni siyang gipulawan. Ako ni siyang gipaningkamot na sige na lang, I hate this online learning, but sige na lang. All right, okay. Para ma-visualize lang. But don't worry, again, I forgot there to mention, these techniques, also this preparation, yeah, we'll make a video out of it. All right, or we'll make a video of them. All right, so don't worry. All right, so you'll not experience the the horror and the excitement of processing stool sana. This is the time na sana talaga that you are to be exposed to the stool, to the beauty and the horror of stool specimens. Okay, anyway. All right, sige na lang. All right. So first is, of course, we need a clean glass slide, all right? And then, of course, label with the pertinent information. You place first a drop of saline, all right? Drop of saline or drop of NSS. So here's our slide. Okay, so with the name, this is my baby. Charot lang. Okay, all right. Yes. So put a dropper, yes. Ayan. 1 drop of your NSS. So that's now your NSS. Quite big. Guys, kani mga drawing ha, are like what I put here na animations. These are just like estimates. This is not like the real size ha. You may be thinking na, sir, it's too big, sir, it's ano. I'm just like estimates lang, no? Rough estimates of the appearances, okay? So it's not necessarily the exact copy, okay, of what you see in the laboratory if you are to perform this. All right, just for visualization purposes. Okay, so don't judge my artistic skills na medyo kulang. Wala talagang artistic skills. Anyway, all right. Ayan, pagpasensya, pasensya na po. Okay, all right. Ayan, so that's your slide with 1 drop of NSS. Next, what you do is using an applicator stick. You then pick about 2 milligrams of feces, enough to form a low cone at the end of the stick, and mix with the NSS. So you mix with the NSS and make sure that the stool sample or the stool specimen are thoroughly comminuted. So when you say comminuted, imong bungkagon, all right? Imo siyang i-mix well. So if you can see na mga naay clumps, if you see like large particles of stool, you need to break that down, okay? Imo siyang i-comminute, okay? Dili commute, charot. Imo siyang bungkagon, all right? Because again, to release the parasites that could have been trapped in those clumps, okay? All right, ayan. So for the procedure, so this is your stool sample. Okay, cutie. Now this is your applicator stick. Now what you do is, diba, you've seen now the container of the stool. So you open, all right? So look at the color, consistency, and all that. Take note of the color, consistency. And then after you get a applicator stick and you stool, yes, go at different areas of the stool, all right? So in a way that would give you a representative sample of the stool because you went through a lot of areas of the stool. So in a way, at least more chances of getting the parasites or obtaining the parasites that could be found in your stool sample, all right? So not only one area of the stool sample, you go at different areas, okay? All right, so as you can see in the drawing, like that, okay? All right, until you form this parang low cone. All right. Again, that's not the real appearance in the real setting. Just again a rough drawing. All right. Now once you achieve this, you then, yes, with the 1 drop of NSS, your slide, you then mix. All right, in the NSS drop. Make sure again that you comminute. Okay, bungkagon. You break up, no, the clumps of stool. Okay, so. Yes, all right. Until you form a homogeneous mixture. All right, it's color brown. Okay, obviously. But depends on the stool, no? All right, okay. Ayan. So that's for the procedure. No, cover with the cover slip and exclude bubbles as much as possible because the bubbles can hinder your examination. All right, so that's your prepared slide already. Okay, the cover slip and then you put. Okay, make sure again that no bubbles are found. Avoid bubbles as much as possible until you have this appearance. Again, this is not the like, you may be thinking, sir, it's too dark or it's too like brown kaayo. No, it's just, again, rough representation of what the smear looks like, all right? But in reality, again, it looks much lighter than this. Okay? All right, so kalma lang dyan. Don't judge me. Okay, I tried my best. All right? Sana na-appreciate nyo. Charad. Kasi, you know, we like to be appreciated. Okay, all right, ayan. So we start first with, of course, we examine under the microscope using your LPO, all right? And then, if you want to confirm the parasite that you have seen, like to visualize the cellular morphology more, you shift to HPO. So you may be thinking, sir, maabutag oil immersion, or like, do we use the oil immersion? No, it's until HPO lang, all right? LPO and HPO lang tayo, guys, for fecal smears or for our direct fecal smear. All right? Why don't we use, or why would we not use the oil immersion? Because under the oil immersion objective, the cellular morphology may be distorted anymore. May be distorted. Okay? All right? So it would be difficult for us to examine because the cell morphology is distorted. Okay? Under the oil immersion. Yes, Mark, what's happening to you? All right, oil immersion. Okay, all right. Now, how do we examine systematically? So, example, you have here the cover slip, ha? So we have two ways. Again, zigzag, you could start from here, all right, like that, all right? Or you could start from here. Okay, ayan. So we have two ways. It's either up, down, or left, right. Okay, zigzag, okay? And then every time you see a parasite, okay, you take note, or if it's an ova, you click, diba, using our counter, all right? Okay, ayan, that's for the procedure. And lastly, report results. Following the one in the previous video, in our previous slides, how do we report the results? Okay, for protozoa, for helminths. Okay, all right. That's for the direct fecal smear. Now, a reminder lang, how will you know that your smear is well prepared? You compare, no, or you use a print, newspaper ba, printed paper, to look if it's too thick or too thin, all right? And it's mentioned there that properly prepared smear, it will allow ordinary newspaper print to be barely legible. Okay, barely legible. So meaning you can't see it, okay, but you can still see the print but not too clear, okay? Like pwede na niya ma-blur konti, alright? But not too thick, okay, na you can't see any print anymore. As long as you can still see a print but medyo blurred na konti, okay? Alright, because too thick make your observation difficult because it's too much stool. There could be a lot of materials already there that are artifacts or confusers. And for too thin, okay, if it's still too thin, yes, you cannot see the parasites or only little. Or maybe you cannot see any parasites at all, okay? All right, so again, not too thick, not too thin. Dapat sakto lang. So not too dax, not too jutes. Okay, all right, ayan, basta smart. All right, now here's an example of a prepared fecal smear. So this is the DFS, guys, ha? This later on is after the D'Antoni. So this is the fecal DFS, okay? All right, now, let's say you want to visualize more, okay? You want to add color. We can use that using temporary stains, especially if we want to see protozoa, right? Especially the cysts, okay? Because it's not necessary to stain your helminth, eggs, and larvae because they don't need that much color, okay? Because they have distinct color naman, all right? But still, they can still be seen if we use this stains, okay? So some of the temporary stains that can be used, mga temporary lang, is your dilute iodine, your MIF solution, di ba? In our lecture in preservatives for stool, merthiolate iodine formaldehyde. There is an iodine there, merthiolate. So it can act as a fixative and as a stain. Okay, so MIF, and you have buffered methylene blue. Okay, so again, these are just temporary stains. Okay, so pang ano lang, temporary lang, like me. Charot. Okay, all right. Again, all right. So again, we'll now focus on the iodine part. Okay, because again, that is what we use. That is now the reagent that we use for the D'Antoni's method. Okay, all right. Because dilute iodine solutions are the most commonly used temporary stains or iodine solutions. Now, what is this purpose? It's most useful for the cysts of your protozoa because the iodine can kill or distort your trophozoites. So, wala na yung pulos. You cannot see anymore the trophozoites of your protozoa, okay? If you use an iodine reagent, you can only visualize the cysts because the cysts can be stained, okay? You can give color to the stains. Now, what's the importance? What's the significance? Why do we need to stain? Because that would help us enhance the nuclei of your cysts, and you can also see the other morphologic features that are clearly seen, that can be clearly seen now, all right? That's why we visualize, or that's why we add stains, especially if you want to identify, in a way, the species of your amoeba. Example, because again, there are some amoeba that look the same morphologically. Example, Entamoeba histolytica and Entamoeba hartmanni, all right? Okay, or Entamoeba coli ba, in a way. So if you want to look at the number of nuclei that is found in the cysts, no, and other morphologic features, then you can add a temporary stain. Okay, all right, ayan. So iodine, again, should be freshly prepared because it can lose their ability to stain rapidly. So it should be freshly prepared. Okay, because again, if dugay na ang iodine, if the iodine has been stored for long, then it doesn't have the ability already to stain. So wala na pulos. Okay, you cannot see anymore the enhanced visualization of your cysts. Okay, all right. And when amoebic cysts are stained, the glycogen takes a dark red-brown color and the cytoplasm a yellowish color. Okay, so here's an example of a picture. You have the cyst in DFS or direct fecal smear, diba? So as you can see, medyo clear naman, but it's still not that clear. So you cannot see that nuclei kaayo. But whereas, ayan, for cysteine diantonis, the one that is pointed, this is the chromatoidal body, di ba? It's much more clearer siya, and the nuclei especially are much clearer. Okay? So we perform this diantonis method or iodine solution. It's usually to visualize the cysts, all right? But again, aside from cysts, it can also stain your helminth eggs, okay? So your helminth eggs will still appear yellowish brown, okay? Because they will take up the iodine stain, okay? All right, ayan. And for the other temporary stains, the MIF, no? MIF is easy to prepare and to use. And use for buffered methylene blue, you need to adjust the pH to be to use properly, all right, so that it can achieve proper staining. Okay. Other methods of the dilute iodine solution, you have O'Connor's and Lugols. Ayan, Lugols. So other types of methods using iodine, still the same, using iodine. All right, the name is just different. Okay, all right, ayan. So these are the other temporary stains and the other methods using dilute iodine solutions. But again, please take note that these are just only temporary stains. Okay, so if you want to just, stain, okay, your stool sample so that you can see the cysts, just for now, okay, for now lang, then you can use these stains. But if you want to preserve, okay, if you want to look at your smears again at a later time in the future, then you cannot use these stains anymore because again, they are just temporary. So after how many hours, the stain will disappear, okay, or the color will disappear. So it will not help you in examination of the smears again in the future. All right, so if you want to preserve for the long run in the future so that you can see again the smear in the future, then we use now other stains, other colors, okay? And that's another topic, another lecture in the later part of the SEM. Okay? Alright. Again, temporary lang to. Alright. Paras ng feelings niya para sa'yo. Ah! Wow, Mark. Wow, that's a hugot. Anyway, alright, drama ka, Mark. Alright. Ayan, now for the method, principle again, protozoan cyst, diba, as mentioned, because the appearance of their cyst is enhanced clearly, and because, again, the iodine acts as the stain. And true enough, guys, this is a board exam question, a recall. The oldest stain, ayan, the oldest stain that is used is your iodine. Nako, please take note, lumalabas sa boards. And this is an advanced question na. This is found in your third year, supposed to be. But, di ba, since we're talking about stains din naman, stains, ang point lang sa stains, guys, is you give color. Alright? Binibigyan mo ng kulay ang buhay ng mga parasites. And not even parasites, also other organisms, especially bacteria. Staining, giving color is very important in bacteria, in your bacteriology, and also in our tissues. When you go to histotechnology, when you process tissues, it's very important, stains. Because again, through stains, we can identify the structures, we can classify them in terms of bacteria, gram stain, gram negative, gram positive, gram negative, yes. And that's the importance of stains, all right? You're just giving color so that it can help you visualize more and to differentiate more, okay? All right, ayan, sige. So again, oldest stain, ako, press the buzzer, that's iodine. Okay, all right. Now, do we see trophozoites here? Yes or no? No, di ba? Kaya namatay na ang trophozoites sa iodine, okay? All right. For the materials, ayan, so as you can see, the D'Antonis reagent is composed of your potassium iodide, iodine crystals, and distilled water. Okay? Of course, other materials similar to your DFS. Okay? And this is the preparation of your iodine reagent or D'Antonis reagent. You dissolve potassium iodide, guys. Ako nang summarize. Sana na-remember pa ang Gen Chem sa mga chemical formulas. Alright? Okay. 1.5 grams of iodine crystals until you reach a reddish-brown color. You filter the solution to remove any undissolved substances, okay? Mga crystals na undissolved. And you keep at the brown glass, a brown bottle, so that it can be protected from light, so that it will not disintegrate, the reagent will not disintegrate, and you store at room temperature, okay? So again, it's not quite important for us to know the preparation because usually when we have our lab activities, it's already prepared. But in the case that you work in the future for reference laboratories, no, for mga other laboratories not in the hospitals, then at least you know, diba? All right. And they have their own methods din naman. All right, okay. And for the procedure, as you can see, it's quite the same. with your DFS. The same procedure with DFS, as in same procedure lang. The difference lang is the use of the reagent. Another reagent, which is your D-antonis. So instead of putting NSS, 1 drop, you put 1 drop of D-antonis. Okay? And then you mix, put the cover slip, examine under the microscope. LPO and HPO. Alright? And for reminders, so the same reminder, allow the newspaper print to be barely legible, okay, through the smear. Alright? Okay, ayan. Okay. And this is now the DFS, and this is now your D-antonis. I'll just repeated the picture. Okay? Alright, so, okay, alright, ayan. So as you can see, diba, you have quite parang darker color, diba, parang reddish brown. This is due to the iodine, alright? So, as you can see, barely legible. You can still see the print, pero not clear, okay? Not to the point na clear pa ang print, ayan. So, barely legible lang. So, this is a properly prepared smear, okay? Alright, make sure again that there are no bubbles. Try to avoid in putting the cover slip, okay? Alright, ayan. Now, for the next video, hopefully, it's the last part na, we'll focus now on the remaining 2 methods, okay? Actually, in your manual, no, these are 2 separate topics, 2 separate activities, but we can perform this as one because, again, they're quite similar procedures lang, all right? Okay, so we'll start with catothic and lastly, the catocats. Okay, all right, so I'll see you on the next video. Okay, all right.